Thank you for joining me for part 1 of my 9th short talk on epigenetic science. In my last talk, we discussed the critical role proteins play in our bodies. If you're enjoying these videos, please like, subscribe, share, and follow. It's very helpful. Thank you. Our bodies are built primarily from proteins, and proteins perform many of the functions in our cells. How many proteins do you think we have in one cell in our body? There are 10 trillion proteins in each human cell. Our bone marrow cells alone produce 100 trillion hemoglobin proteins per second. So how do our cells make proteins? Our bone marrow has to produce so much hemoglobin because we're making 2 to 3 million red blood cells every second. During this short video, we'll make somewhere around 1.5 billion red blood cells. In fact, proteins are being made in many of our cells right now. It's a fascinating process, and it's important we have at least a basic understanding of how our genes are expressed into proteins as we continue to deepen our understanding of epigenetics. My next few talks are going to be a little more scientific in terms of how our genes are actually expressed. If it seems like a lot of science, I'm doing this to give us a solid foundation in the power of epigenetics. It's not something we have to completely understand in order to successfully apply epigenetics. So please enjoy. My name is Robert Needleman. I'm a molecular biologist and biochemist. Thank you for joining me for this episode of Empower My DNA. We know from our last talk that proteins are chains of amino acids. What's incredible is the genetic code in our DNA holds the information to make proteins. The question is, how do we start with a DNA molecule and make something as complex as a protein? In Talk 5, we discussed the structure of our DNA, or deoxyribonucleic acid. This is from the MIT X Bio. DNA is double-stranded, or dsDNA, and the two strands are interwoven to form a double helix. On the inner part of the double helix are nucleotide base pairs. A always pairs with T, and C always pairs with G. This means our DNA is very long strands of the letters A, T, C, and G. But what do these 4 letters mean? In 1968, a Nobel Prize was awarded to these 3 scientists for their interpretation of the genetic code and its function in protein synthesis. They figured out that different combinations of 3 letters in the DNA hold the code for each of the amino acids. For example, A, T, G in the DNA are the letters for the amino acid methionine, and the letters C, A, G are the letters for the amino acid glutamine. So a gene for a protein is actually groups of 3 letters called codons that define the amino acid sequence of the protein. It's really quite profound. Now, if we look at a representation of a cell and we look at the parts of the cell, we see that cells have a cell membrane which separates them from their surroundings. Inside the cell, we have a gelatinous fluid called the cytoplasm, and if we continue inside the cell, we come to the nucleus, which houses and protects our DNA. The nucleus is separated from the cytoplasm by its own membrane, which also isolates the DNA. Our proteins are actually made in the cytoplasm, so the question becomes, how do we get the instructions from a gene in our DNA, which has to stay in the nucleus, to the cytoplasm so the gene can be made into a protein? To help us understand this amazing process, I'm going to use an analogy of baked goods being made in a bakery. In this analogy, the bakery represents the cell. All the recipes for the baked goods represent the DNA in the cell. The baked goods represent the proteins the cell needs to make. In a bakery, the work that goes on in the office is as important as the baking. The recipes are stored in computers in the office, separate from the kitchen, to protect them and keep them safe from being damaged. This is similar to our DNA being housed in the nucleus. In order for a recipe or gene to be used, a copy of the recipe has to be made. The copy has to be delivered to the kitchen, the cytoplasm, where the bakers and ingredients are located. The recipe can then be used to make the baked goods or proteins. A similar scenario takes place in our cells. A gene in our DNA must be copied in the nucleus into an exact copy called a messenger RNA or mRNA. This important process is called transcription. Transcribing a gene produces an mRNA copy of that gene. Here is RNA side by side with DNA. RNA is similar to DNA with some very important differences. DNA is a double helix and uses 4 bases, C, G, A, and T. RNA is a nucleic acid like DNA, but it's single-stranded, which allows it to fold into many different shapes. RNA uses 4 bases, C, G, A, and the T is replaced by U. We're going to go through an exciting, simplified version of transcription. When a gene needs to be expressed, an enzyme called RNA polymerase attaches to the DNA at the beginning of the gene. A gene carries the code for the sequence of amino acids in a protein. Here's the highlight of a segment of the gene. Within the gene, the specific order of the bases dictates the order of amino acids that will make up the protein. Each group of 3 consecutive bases is the genetic code for a particular amino acid. As a result, each group is referred to as a codon. RNA polymerase causes the beginning of the gene to unwind the double helix. One of the strands is called the template strand, and it will be read or transcribed into messenger RNA. The other strand of DNA is called the non-template strand, and it is not transcribed. RNA polymerase uses the base pairing rules to assemble free nucleotides in the nucleus into a strand of messenger RNA that is complementary to the template strand. For example, RNA polymerase reads T for thymine and then binds it to free A or adenine. This process continues with C binding to G and G binding to C. Whenever RNA polymerase sees A on the DNA template as it's building the RNA, it pairs A with uracil or U instead of T. As this process continues, the genetic code for the gene in the non-template strand is transcribed into the mRNA, and we have an exact copy of the gene. Here's a diagram of the genetic code for the 20 main amino acids. The 3-letter codons are all described by their first letter, second letter, and third letter. You see U in the 3-letter codons because the code is normally described in terms of the messenger RNA. The way we read the chart is the codon CCU codes for the amino acid proline, and so on for all the amino acids. What an amazingly simple and profound way our cells store information in our DNA. Before we go, I have an incredible real-time animation of the process of transcription from the DNA Learning Center. Here is a strand of DNA with a gene that is going to be expressed. The first step in the process is the binding of transcription factors. At the beginning of the gene is a promoter region. This is where the transcription factors attach. In this case, the gene runs to the left along the DNA following the promoter. A mediator enzyme complex arrives carrying the enzyme RNA polymerase, which is shown here in green. Other protein factors arrive as well to help with the start of transcription. These proteins work together with RNA polymerase. The assembled collection of all these factors is referred to as the transcription initiation complex. And now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific regions of DNA known as enhancer regions that can be thousands of base pairs away. Contact between the activator proteins and the initiation complex releases the copying mechanism. I paused the video so you could see the DNA, the RNA polymerase, and the messenger RNA being made. RNA polymerase is copying the template strand of the gene into a messenger RNA. Here we see RNA polymerase has unwound the DNA double helix so that it can transcribe the gene. Here we can see the many free nucleotides in the nucleus that the RNA polymerase is using to build this messenger RNA. And here you can see the actual intake area where the G, C, A, and U nucleotides enter. And here we see the completion of this magnificent process, creating an mRNA copy of the gene. Was that incredible to see? Our cells are awesome. So after the mRNA is made in the nucleus, like we just saw, it goes through several forms of editing before it then leaves the nucleus to be made into a protein in the cytoplasm. In part 2, we're going to explore how the messenger RNA is read into a protein in a process called translation. Through epigenetics, we are truly in charge of our health and our life. Understanding how our cells express our genes deepens our belief in our empowerment. Thank you very much for joining me. I look forward to seeing everyone on part 2. Have a most incredible day. Take good care.